Yeah. So now I'd like to take the opportunity to introduce our speaker. Dr. Philip Dykema is currently serving as the Deputy Director of the Microbiology Section of the Washington State Public Health Laboratories, where he monitors current testing to maintain compliance with Washington DOH accreditation agencies and assists in onboarding new test methods, including expanding the use of whole genome sequencing to monitor reportable diseases. Prior to serving at that lab, he worked for 10 years at the National Veterinary Services Laboratory in the Mycobacterium and Brucella section, and worked for 6 years at a developmental laboratory for an agricultural company. In 1997, he received a PhD degree from Iowa State University in molecular, cellular, and developmental biology. And he's going to be talking to us today about rolling out whole genome sequencing at the Washington State Lab, which I think a lot of us have been looking forward to, and we're very interested to hear all about it. So Philip, please take it away, and thank you for speaking.
All right, can everyone hear me, and can everyone see the slides? Fantastic.
Yes,
all right.
Yes, yeah, you're good to go.
I want to thank everyone for inviting me to speak here as you celebrate World TB Day. Something that came out, if you heard the introduction, I worked at National Veterinary Services Laboratories for 10 years looking at tuberculosis and brucella in animals. And then from there, I found my way over to Washington Public Health Laboratory to work as the lead in the TB lab here. And so I met a number of you in that capacity as I served there, and then slowly worked my way up into the position that I have right now. And so it's an honor here to stand and talk to you a little bit about something that has been a slow train coming. We're trying to get it down the road, and we're finally got here. And we're gone live with whole genome sequencing here for TB at Washington Public Health Laboratories. So some of the takeaways I'd like you to have when we leave is just understanding how genetic mutations can impact drug resistances as you administer antibiotic therapy and what that really means. We're going to kind of dig into the biology a little bit, not real deep. We're not going to give you a quiz. You're not going back to college. We're just giving you an understanding so you have a placement as to when I'm talking about mutations in genes, what it means for drug treatments. And also, I want you to understand how we're integrating whole genome sequencing into our public health TB lab workflow and what that means and how we're moving forward into the future. Who's doing all of this work? Well, on the left-hand side of the slide, you'll be able to see the TB laboratory, and many people have attended the TB echo sessions as well as some of the other interactions you've had over time. In the middle slide is a group of people that you may not be acquainted with. There are Advanced Molecular Detection Laboratory. They actually do the hands-on sequencing. They prep up all the genomes, they get it onto the sequencing instruments, and they help get it up into the cloud once the reads are done. And on the right-hand side is the bioinformatician, Kristen Waterman, and her companion animal, Cricket. And she has established the pipeline that we're using at the moment, and also setting up those new reports that you're seeing come out of the laboratory. So, like I said, today we're going to talk a little bit about a brief review of the biology principles that surround mutations and what it means for drug therapy. We're going to give you an overview of our validation, how it turned out, what we looked at, what we established as ground truth for ourselves, and then how we're established our Washington disease report, as well as how we're integrating into our workflow. So a review of those biological principles. DNA. It starts with right here at the deoxyribonucleic acids, and which is really a complex molecule that's composed of two subunits, a sugar phosphate backbone on which are sitting nucleotides. Now, I've had a little bit of feedback already, and the colleague that shared that said, are they really nucleotides or are they nucleosides with an S-I-D-E-S? And the honest truth, it's a nucleoside. When it's bound to the sugar and phosphate background, that's when it becomes the nucleotide. So yeah, the jargon in the field confuses the two, and I'm guilty. I am guilty of it. And there are 4 different types: an adenine, a guanosine, a thymine, and a cytosine. Don't need to remember all that. It's just that there's 4 different types that bind themselves together as they wrap themselves up in the DNA helix. How does it work? DNA is the basis for all the genetic work, and through a process known as transcription, it flips itself over to RNA. The only difference between DNA and RNA is that sugar backbone. There's just a little tiny portion of that backbone that makes a difference. And what it does is it establishes that the RNA is much more labile. It goes away really fast. And that's because there are enzymes out there that are chewing away and making sure the RNA doesn't stick around. Biologically, there's some hypotheses. What does it mean for us as organisms? A lot of viruses are RNA. That could be why we have it's labile, all the rest of it. It's a hypothesis. There's nothing established there. It's just the process of going from DNA to RNA. And then there's a second step, from RNA into proteins. And that's the biggest step right there. That's when we go from the nucleoside-nucleotide coding to the protein, the amino acids that are all lined up in a row, and you get to a protein. What does it mean for drug interactions? Those drugs can slip into pockets, interact with the protein in a way that then changes the configuration of the protein and stop its function, which is really important, because if you want to stop the bacteria, you want to be able to stop its ability to form a cell wall. You want to stop its ability to use nutrients to continue growing. You want to be able to interrupt its ability in some fashion and or form. That's what the drug therapy is all about. Now that puts the organism under a lot of pressure. And biology has this interesting way of mutating the DNA sequence over time through a process known as replication. It's not a perfect system. The replication of the DNA over time is not perfect by any means. And so we introduce, happens is introductions of mutations happen. We call them single nucleotide polymorphisms. One of those bases changes. It goes from adenine, say, over to thymine. It goes from cytosine over to guanosine. And it just happens over time. Some other things that can happen, you get insertions, little pieces of DNA gets inserted in, or get plucked out, and you get eliminations. And so all those things affect the coding region. Remember, we're going from the DNA to the RNA to the protein, and that affects the ability to create a protein. So what does that mean? So I'm just wanting to focus on this complex slide right here on the left-hand side. So say in the whole process of it, there is alanine, which has the ability to do 4 codons. Now that's because when you have an RNA, it comes off the DNA, you have an RNA, there's a little leader sequence where the instructions of the machinery comes along and it stops and it says, okay, here's the start. And it's interesting in biology that start is always a methionine. After the methionine, then you begin 3 amino acids, 3 nucleotides, 3 nucleotides, 3 nucleotides to form an amino acid. Alanine has 4 different possible 3 nucleotide codons. And I'm just going to point to the one that I've highlighted in red, the GCC. It codes for an alanine. If a point mutation, a single base mutation, would change, say, that second one to a G, it flips itself over to a glycine. What does that do? It can affect the shape of the protein. So if you go to the bottom and look at the left-hand side, there's our original protein. It may shape it. It may squeeze it together. It may change that pocket somehow so the drug can't go in and interact. That's a valuable asset for the bacteria to continue to be able to grow. It can get away from the drug and continue to use that protein. Alternatively, there's also the ability to stop translation, and that happens through 3 codons, 3 nucleotides together, and they're at the bottom of the screen here next to the word stop. What happens if, say, a leucine, which has the ability has 6 different codons, and that TTA mutates, so the second one flips over to an A? There's a stop. It also automatically, the machinery comes along, it finds it, it stops the translation of it, and the protein then flops off the machinery. You get a truncated, like on the bottom right-hand side here, you get a truncated protein somehow. It could still be functional. And that still allows the bacteria, because the pocket is gone, the bacteria can still use that protein and move forward. Now, all of these mutations are not advantageous to the bacteria. by and large, all these mutations is just a random card, random happenings here that over time, a bacteria could come up with something. Often they're silent. These mutations are silent, which means that it doesn't change the protein at all. A lot of times they're deleterious. So in other words, that mutation has caused the problems in the bacteria. They can no longer use that protein, okay? So there's a whole wide variety here, but the whole game in nature is to try to get away from the selective pressure. And when you apply a drug therapy to a patient, you're applying pressure to the bacteria to try to move away from it and introduce resistances. So that's a wild, crazy ride through the world of biology, and just a reminder of things that you kind of knew already. The overview of our validation. What did we do? How did we go about proving to ourselves that our whole genome sequencing workflow was going to be concrete and valid enough to send out reports that affect you as clinicians to treat your patients? Okay. We chose 93 isolates showing a wide variety of drug susceptibilities, pan-sensitive, single drug resistances, multi-drug resistances in the same genome. Now, we worked with CDC to get the whole genome sequencing results and compare. So what we did was we chose isolates we've already sent to CDC that got a genotype. So you guys got that back already. You understand the genotyping process that CDC goes through. We chose only those genomes to grow up here and sequence through. We asked CDC to reach up into their databases and pull down the genetic sequencing that they did and analyze it for those 19 genes. And they're all right there. You don't need to worry about the whole specificity here. And they did it and ran it through a pipeline. We did it and ran it through the very same pipeline. So Kristen worked with CDC to bring over a pipeline that they were using as they did whole genome sequencing antibiotic resistance detections. And we installed it here, got it up and working, proved it to ourselves that it worked properly, and then began our validation with them side by side. So we looked at two different things. We looked first at the mutations. So were we able to see mutations that CDC saw? And if not, what kind of error rate did we have there? And the interpretations. So we related it back to the drug. Did the mutation we saw have an effect on resistances, known resistances, okay? We also looked for, can we do it day after day after day and get the same result? And we created this wonderful report. So let's dig in a little bit and look at those mutations. So on the left-hand side here is the list of the drugs. And in the second-hand column there is a list of all the genes that are associated with that drug and known resistance types. And then next to that is the sensitivity results that we got, comparing our results with CDC results. And literally, sensitivity is looking at for false negatives. Did we say there was no mutation and CDC said there was a mutation? And the answer was no. We saw everything that CDC saw, and everything that the CDC said there was no mutations at those sites, we said the very same thing. Specificity is looking at false positives. We said there was a mutation that caused a resistance, and CDC said there was nothing there. That's all perfectly normal. That's an equal genome. It's a wild type genome. You can see here we had two of them, one in the AHPC area, as well as one in the F-ambutal area. And we're going to talk about that in just a few minutes. We were very excited about these results, by the way, because what it said to us was our sequencing workflow is equaling what other people in the field are doing, and we can now share our sequences as well as compare it to what other people are doing. Very, very excited here. So this is another way of looking at it, and it has to do with the interpretations. Now, I'm going to pause for a minute because one of the feedbacks came back, why aren't you looking at IPHC and including it in the interpretations? And it has to do with that pipeline that we borrowed from CDC and brought here. That particular workflow, that particular bioinformatic workflow, looks at only these 5 drugs and known mutations in specific genes of those drugs and doesn't include all of them. We just discovered this recently, and we know it now to be a limitation of our workflow. We are looking for ways, and over time, We're going to be steady here. We've got something up and running. We're going to be able to answer to some of this stuff, but over time, we're going to be able to look at other additional genetic genes associated with resistances to various drugs, okay, including bedaquiline. We're not going to stop there. We're going to introduce that over time, but we have to be very careful and bring in the bioinformatic workflow and validate it carefully. All right, back to the interpretations. How did we do? So this is just a way of looking at possible interpretations, susceptible, resistance, or unknown. And minor errors, when we said it was unknown, CDC said it was susceptible. Unknown simply means the catalogs and what we're trying to compare to. It's unknown whether that mutation works or not. We'll get back to that again in a minute. Major error is we called it resistant. CDC said it was susceptible. And a very major error is we said, oh, nothing there. And CDC goes, there was something there. Sorry. And as you can see, major and very major errors, we did not. We were spot on, walking hand in hand with CDC, saying, well, if they said it was there, we saw it as well. Those minor errors had to do with unknowns. And occasionally this happens for ourselves, that we calling up an unknown and CDC is going, it's susceptible or it's resistant, and we say unknown. We put this into our SOP and locked it in. So here's what we're going to be doing. We're going to be doing side by side. We're not getting rid of antibiotic sensitivity testing. We're going to have whole genome sequencing side by side with our whole genome sequencing side by side with antibiotic sensitivity testing. And we're not going to do away with it for now, so that we don't get into a situation where we told you something and the resistance types are phenotypically resistant or susceptible. How do we integrate it in? Here's the report. Some of you have seen it already. I know it's a lot of work on this one. We're going to dig into it, so don't worry about it. We've chosen this particular format because it fit the output from our bioinformatic workflow. We have had requests, can you make it look more like the CDC old MDDR report? And while we'd love to do something like that, that's CDC's report. And we're going to walk through it today with you so as to get you more familiar with this and comfortable here with this particular report. One thing you'll notice here is that there's nothing in here that talks about percentages of this mutation is seen in so much percentages of the genomes in the area. We don't have that capability. We can't do that kind of reporting to you right now. As a CLIA and CAP regulated institution, we have to abide by their rules and regulations, and we just don't have the backup of known genomic information to say yes or no on percentages of genomes out there. So let's dig into that upper portion of it. By CAP regulations, we have to put all this on there. What is the report? Who's doing it? That's us, the Washington State Public Health Labs, our address, our contact information, the data that we created the report, the weight, the sample that was sent in, the version of the pipeline, all of this is important. Of course, the patient name, birth date, collection date for that sample will be on there. Where did it come from? Who's getting the information and who submitted it? And this is the meat. This is the meat of the report that we're giving to you. On the left-hand side is the drug. So EMB, ethambutol, fluoroquinolones, isoniazid, PZA, pyrazidamide, and rifampin. The drug result is what we saw. So EMB-B, that's the gene, and in that gene we saw a change in the protein at their amino acid 378, going from a glutamate to an alanine. Okay, the interpretation. The interpretive software that we have linked up with our pipeline, our workflow, says that that is susceptible. It's not known to confer resistance. Okay, we can page ourselves down fluoroquinolones. Nothing was seen there. Let's page ourselves forward down to the pyrazidamide. Oh, there was then the PNCA. That's the gene at amino acid 57. We went from a histidine to an asparagine. Aha, that is known to be a known phenotype or known mutation that can cause resistances. And so this mutation is expected to show resistance against the pyrazidamide drug. Now, what isn't shown here is the unknown. And so I have it down below here as what it would look like. Again, the drug, it could be any one of the drugs, the gene that's associated with it, the change in the amino acid, the interpretation, there's going to be a U, okay? And the effect of that mutation on pyrazidamide resistance is unknown, okay? And so that's a matter of, okay, once I have that, I have to wait for my antibiotic sensitivity testing to come out from the laboratory. And then finally have a long discussion about what is in the report, how it was generated, where are we getting our information from. Literally, it's about we're comparing ourselves to the WHO catalog and the CDC collections here. And what's important here is that we adapted it for ourselves. It's not an FDA-approved test by any means. Even though we obtained a lot of the workflow from CDC, this is our work. This is our own laboratory-developed test. And highlighted here, I can't emphasize it enough. These results are for us an infection control. Please do not use it as the sole means of diagnosis and patient management. Use it in combination with other things, but you know those things already, don't you? You've already known it with acid fast. You wait for the PCR to come back to you, don't you? Because those acid fast may not be TB. It could be a Mycobacterium avium complex. So again, just use careful, thoughtful ways of how you're going to handle this information as it's moving forward. Here's our workflow. You're going to send samples to us. We're going to process it. You're always going to get a smear. You're going to get a gene expert. You're going to get culture from us. Any positives, we're going to put through PCR. You're also, if it's a first time identification of this patient, you're going to get drug line susceptibility testing, both liquid and solid if you ask for it. And there it is on the right-hand side. You're going to get a whole genome sequencing result from us. Let's take a look a little deeper at that second line testing. The turnaround time for drug susceptibility testing is about 3 weeks. The turnaround time for our whole genome sequencing is 1 week. Literally what we're doing is we're starting the sequences in the laboratory on a Friday, and often by Wednesday the next week, those results are in your hands. So once we've identified an isolate, the pure isolate, we can bring up whole genome sequencing for you immediately. Now, pyrazidamide has been discontinued in our laboratory for various reasons, and it has to do with the confidence we have in the drug susceptibility testing of it. And that's nationwide. Now, if we observe potential resistances and unknown determinations, which we can't support with lab tests, say that pyrazidamide test, the isolate's going to be sent to a reference laboratory. Our reference laboratory is Wadsworth. And so within another week, we can turn it around and say yes or no, that pyrazidamide unknown or that pyrazidamide resistance, that's confirmed. Now, again, I mentioned to you that both the drug determination of resistances and the lab testing of the drug determination are going to continue, okay, side by side. And I emphasize this here one more time because what I'm going to be doing is as we move into the next few months, I'm going to open up what's called an open invalidation. And my idea here is this. I'm going to be doing drug susceptibility testing collation next to the genotype AMR testing that we're doing side by side. And what I'm looking to do is gain confidence that when we say pan-sensitive, it is pan-sensitive in the phenotypic testing. My magic number is 700 isolates. 700 different patients. I have to do it that way, 700 different patients. So this is going to take time in the state of Washington, where we see about 250 a year. So over a few years, we're going to be able to finish this off. And my goal is this. My whole goal is this. Not to stop. If we see unknowns and resistances, I want to have phenotypic testing on it. But if we say pan-sensitive, I want to stop with confidence and say it's pan-sensitive, and we will no longer test. So we will come up with the genetic, at the end of the validation, we'll be able to say, if it's pan-sensitive by genotyping, we stop. And we're not going to give you any phenotypic testing because we're standing here with confidence saying it's pan-sensitive. And with that, I'm going to stop and I'm going to take questions and hopefully have a wonderful interaction with you all.